my name is Mallak Ali. I'm a research associate at Chan Zuckerberg Imaging Institute, and today I'll be presenting about how we've used specifically JCVI-syn3A cell line to assist in automating the cryo-electron tomography pipeline. Okay, so to orient ourselves a little bit, I want to give a very brief introduction to cryo-EM overall for the folks that may be a little more new to it. So it's a re-emerging structural technique from the 80s that's become increasingly more popular as more tools are being developed to work with the cryo-EM pipelines. So there are basically two paths that one can take in cryo-EM. There's single particle cryo-EM or there's cryo-ET. So single particle assists in achieving very high resolution structures of macromolecules and their associated complexes. So here's a structure of eubacterial and eukaryotic BTPases reconstructed by Noguchi and Yavursky from the Molecular Medicine Department of the University of South Florida, where we can see very high resolution alpha helices and all their intricacies, as well as the transmembrane domain. But I won't discuss this too much because the real star of the show and the star of Chan Zuckerberg Imaging Institute is cryo-ET. So before jumping into it, I just want to provide an example of what's achievable using cryo-ET. So here's a very beautiful image of mouse sperm and axoneme, where we can very clearly visualize the central pair as well as the surrounding doublet microtubules. What's especially cool about this reconstruction is that it's actually on the CZII cryo data portal, which I'll touch on more later. So in comparison to other microscopy techniques, cryo-EM is becoming more favorable in the sense that the sample prep is less complex and it can provide very high resolution 3D images for varying biomolecules without eradicating the sample in the process. So in the case of cryo-ET, we're able to view the complexes in situ, and since the cells are flash frozen in their close to native state, they can give researchers a relatively accurate depiction of what's going on inside the cells. So while NMR and X-ray crystallography are capable of reaching 1 angstrom resolution, they don't capture the cellular ultra-context structures in their near-native states. So in the context of JCVI-syn3A, it falls well within the range of cryo-ET, which makes it very nice for our purposes. So what exactly is cryo-ET? I've told you about its results and how it compares to other techniques, but how do we go about using cryo-electron tomography? The gist of it is that cryo-ET is a fast-freezing biological sample in their close to native state for the purpose of achieving near atomic 3D resolution of the sample of interest. So the main difference between a single particle analysis and cryo-ET is that single particle analysis or single particle cryo-EM captures the particle of interest at various orientations and then averages those tomograms to create a 3D reconstruction. But cryo-ET captures a series of 2D images called a tilt series that aids in the 3D reconstruction of the particle. So this is a general schematic of what the process looks like, and for the most part, it's pretty straightforward, but the current bottleneck is in data processing, which is where JCVI-syn3A comes in. So the great thing about JCVI-syn3A is that the cell's diameter would typically average at about 400 nanometers, meaning that we can bypass fib milling. So fib milling is a focused ion beam milling that's required when the sample is too thick. So as the name suggests, it's a cluster of focused ion beams that precisely etch away at the layers of the sample. So it's very time consuming, very costly. So it's nice that we can avoid that with syn3A. And it's derived from mycoplasma mycoids. It's already pretty small, considering that they lack a cell wall. And for comparison's sake, next to E. coli, that would definitely require fib milling, and we would have to freeze it on the high pressure freezer. So it'd be a different protocol altogether. So syn3A is very nice for our purposes. So moving forward to sample preparation. So based off of previous literature, we cultured the cells in sp4-KO media until they reached a log phase, which we measured on the nanodrop using the optical density. And then we glow discharged quantifoil copper 200 mesh 2-1 grids, applied 4 microliters of the syn3A pellet, 6 seconds of blotting, and then plunge froze directly into liquid ethane, and then migrated it over into liquid nitrogen for storage purposes. So as I mentioned before, the cryo-ET is capturing images of the same region of the grid at different angles, and altogether, the tilt series received a dosage level of about 120 electrons per angstrom squared. And for those that are familiar with cryo-ET, this is a little higher on the range, but higher dosage level results in increased signal-to-noise ratio, which means better contrast for our tomograms. But if you're unfamiliar with the sample, you can do a series of burn tests through like empty regions of the grids to see what it is that your sample can take. And then we collected a tilt series of, or a tilt range of 45 degrees. You can go from anywhere from 45 degrees to 60 degrees, depending on your sample. So from this, Reza, who curated the data collection, was able to collect more than 800 tomograms over 4 data sets. So from data collection, we go into data processing. So I'm not going to touch on all these details considering time, but I will touch on the most important parts for the presentation today. So we touched on the tilt series, which was collected during the data collection, and then moving on to our TOMO3. So many issues can arise during data collection, and one of which is beam-induced motion, which is what our TOMO3 collects or corrects for, apologies. So those that are familiar with cryo-ET, you may have heard of Motion Core 3 or TOMO2. So our TOMO3 is a new reinvented version. And it stands for Alignment and Reconstruction of Electron Tomography, or for electron tomography. And it allows for real-time automated reconstruction of the cryo-ET tomograms alongside data collection process that collects iteratively with each new tilt by way of GPU acceleration. So with each new tilt series, the software corrects possible faults in the data correction process. So that can include the beam-induced motion, it can estimate the contrast transfer function, realigning the tomogram, and allowing for the 2D reconstruction of the sample. So we're also using the mini cells for QC purposes. So it's used to help train our TOMO3. So what's happening is that of the 800 or so tomograms that were collected, only 265 moved forward in the processing pipeline because our TOMO3 was able to recognize the well-aligned, the very visible individual ribosomes, and then rejected the rest of them. So it's very nice for optimizing the pipeline and making sure that, bad tomograms don't go forward with it. So as we can see here, we can clearly see the ribosome, but there can still stand to be some optimization. So then we send it through denoise-ET. Denoise-ET is an in-house algorithm created on a noise-to-noise basis, and it increases the signal-to-noise ratio of the tomograms after our TOMO3 processing. So it cleans up the image, increases the contrast of the molecules of interest, so we can very clearly see the ribosomes that emerge as we go through the Z-slab of the sample. Moving forward in the data processing pipeline, we then go through manual picking, which I won't touch on considering that today's presentation is all about automation. So I'll move forward into mini slab curation. So mini slab curation is also called 2D averaging and classification, where we take multiple slices of the tomogram and then project them into 2D images. So it turns a 3D issue into a 2D issue, where we can get gallery images to see if there's variation in the processing. So these are automated picks that we're then able to observe, and is integrated with the CryoSpark. So CZII pipeline integrated with CryoSpark, and allows us to visually inspect picks and curate a set of micrographs. So we're also using this for quality checks, and this diagram was provided by UA, and it beautifully illustrates the 265 tomograms that were accepted. Of that, 591,000 picks were chosen, but you can see that they're not too great. So then by way of mini slab curation, 19.8 thousand picks were selected, and moving forward after overlapping the slabs, only 16.8 thousand picks will move forward in the processing pipeline. So those 16.8 thousand picks and 265 denoised tomograms will move forward and will be posted on the cryo-ET data portal, which is what I mentioned earlier. So we'll be submitting those relatively soon. And a little quick intro to the cryo-ET data portal. It's a beautiful, well-crafted platform that helps bring together structural biologists as well as machine learners onto this platform, and basically uploading your tomograms, you can upload them raw, and then it'll send it through this entire pipeline. It's automated, so then you can get out your 3D structure. And it's really nice, very easy to use. I used it myself, and I don't have much computational background, but it was very easy. And we really encourage everyone to upload whatever data sets they're comfortable with uploading to the cryo-ET data portal and share for public access. With that being said, that's all to my presentation. Thank you so much again. Thank you so much again for listening, and I want to thank everyone at CZII. While I presented their work, everyone was super crucial to the pipeline and did amazing work, especially Reza and Yue, that were really instrumental in really optimizing the cryo-ET portal. And thank you again to JCVI for giving me this time. Thank you.
Thank you, Reza. Mallak. So this is technology that I think is really new to most of the members of our consortium. And so I know that we have previously, through cryotomography done with Elizabeth Villa at UCSD, been able to, for instance, count the ribosomes in the cell, and then the computational biologist at the University of Illinois made an estimate, so tried to thread a genome between the ribosomes to get an idea of its structure and location in the cell. With this data set, what else do you anticipate that we might be able to learn? Can we identify, how practical is it to identify other proteins or protein complexes in our cells?
I think the aim is that so syn3A just was especially ribosomally dense. So we used it for those training purposes, but currently at CZII, we're training the algorithms to recognize a lot of different protein complexes, aiming for the larger ones as of right now. But the goal is that eventually the more common protein complexes, they should be recognizable by these different softwares and should be able to send it through the pipeline just to be able to recognize. And you don't have to go through the manual process of picking all these individually.
And so go ahead, James, do you have a question?
Oh, just beautiful talk. And I had a question about the sample preparation. So in the cryo-electron tomograms that John mentioned from the BIA lab, I think the cells during blotting were squished to a depth of approximately 160 nanometers. Is that true for the current sample preparation as well? Are there ways you can imagine to retain the native shape of the cell somehow? And again, beautiful work.
Thank you. I think we had a similar experience in which the cells were squished as well. Although, as I mentioned, that was kind of optimal for our purposes, considering that we didn't want to fib mill. But I would assume that you can also decrease blotting time to sort of retain the natural thickness of the cell, but I'm not completely sure.
Thank you very much.
I like the squishing process. So is that, if we assume that a spherical membrane will be the maximum volume that the cell can hold, if you squeeze it, either the membrane has to expand or you've got to lose some volume. Any thoughts about that?
I'm not completely sure because we didn't have like issues with that. I would say it would we would have to go back to the sample preparation and then optimize on that protocol if that was seeming to be an issue. I think otherwise before plunge freezing, we can decrease the blotting time to not have it so like squished. Other than that, I can't think of anything, but I will try and maybe discuss it and then get back to you as well.
Okay. Other questions? James Sayens, how much can a membrane stretch?
Good question. Typical textbook value is somewhere around 3 to 5% area. Not much.
Okay, that's useful to know.
Yeah.
I'll be back in touch with you about this, Mallak. I'm really curious.
Thank you.